One.
Hello, Casey.
Hi, everybody. Nice to meet you for the first time.
I still accepting participants in the conference room. Casey, just for you to know.
Oh, thank you so much for joining us for this new edition of the 2060 longevity investment pitches. We've got two really exciting pitches today. Whilst people are joining, I'll just do a very quick intro for people who haven't been here before. I'm Cassie. I joined the 2060 forum. The 2060 Foundation back in January to help Gabrielle with organizing some of these pitches and also the 2060 Longevity Forum. So today, as I said, we'll just very briefly do a short introduction, and then for those of you who haven't been here before, we'll do two pitches, 10 minutes each, followed by 10 minutes of questions. So we've got Myworkout.ai and Cyclarity today. And let me just check that everyone is in the call. Yes. So yeah, the 2060 Foundation is a nonprofit. Our purpose is essentially to defeat aging by 2060. And this is through a bunch of various initiatives, including the pitches to bring more capital into the field. And so this is one of the initiatives. We've got the 2060 forum happening later this year. Please let us know if you want more information about that. It's going to be a conference taking place at the end of August in Aix-en-Provence. Beautiful place and with a bunch of really cool people invited. So if you're an investor or if you are interested in longevity, reach out so we can give you more information on that. Just a very quick overview of our team here. A bunch of different skills, and I think we're really well suited to kind of push this forward in the future. So we've got the first pitch, which is Myworkout.ai, and I will stop sharing very quickly to let Scott, if you would like to share your slides and do a little quick introduction on yourself as well.
That'd be great. Thanks, Casey. Great to meet everyone. You're going to see we're actually going through a little bit of rebranding. So we actually are under a new name, MoveWell AI, with a lot of the same of everything we're doing. So you'll see something a little different. Can everyone see my screen? Cool. All right. I'm going to hide you guys and just kind of dive into it. So let me ask you a question to kick us off. Did you move today? Because see, movement is medicine. Every step, every rep, every day. So great to meet everyone. I'm Scott Finkelstein. I'm the founder of MoveWell AI, and I've accomplished some amazing things in my career. I've led companies to over $6 billion in new revenue. I've scaled global tech, coached C-suites, and built teams. But this MoveWell AI, this is different. This is the most important thing I've ever done. It's my calling. It's my purpose. See, humanity deserves better. And the current state does not give us better. It waits until you're broken so it can profit from us, from our pain. And today, we shift the paradigm. We take back humanity. This is personal. And I'm going all in. So the problem, simply, two worlds in one broken system. See, medicine and fitness, they don't talk. They exist in silos and even silos within themselves. Trainers aren't trained in chronic disease. Doctors don't prescribe movement, and the people stuck in between, well, we all suffer. We have a $4.5 trillion health economy, And not a single system uses movement, the most foundational function of life, as a clinical tool. That ends today. So we've created the solution. It's called movement intelligence. We've built an AI system that learns how you move, predicts what's coming, and adapts in real time to prescribe the exact movement your body needs. It's not just tracking steps, it's building clinical grade protocols for injury prevention, rehab, and long-term vitality. This is what happens when you fuse medical insight and functional movement and adaptive AI into one platform. Every movement becomes medicine. Every step becomes strength. How our technology works. And here's how it's powered. So we've got intelligent inputs from wearable devices, camera capture, and clinical baselines. We've got real-time AI adaptation. Responds like a trainer, moves like a physical therapist. Medical grade logic built in from day one. So it's the smartest physician in your life. Personalized evolution. The more you move, the smarter it gets. The AI constantly is adapting how it's helping you move better. This isn't another platform. This is an operating system as sophisticated as the human body, but built for longevity. Now, what we know is we got a $4.5 trillion opportunity here, right? You think about it. Digital health, $660 billion. The longevity market itself, and that's what we're here for today, right? $1 trillion. Musculoskeletal chronic disease is 1.4 trillion itself. Then you dive into early adopters are hungry, aging boomers, biohackers, self-insured employers, digital health clinics. We have a huge market that is thirsting for what we're doing. And as you look at how spending is shifting from reactive care to predictive health, that's where MoveWell AI sits. At the center of humanity's next evolution, looking to serve a global demand for smarter prevention, better recovery, and for all of us, a longer life. And not just a longer life, but a longer life where you're going to feel good living. So when we look at our go-to-market, from consumer to clinical, a strategy that scales across all of health. Each phase expands our impact and our intelligence. See, we didn't wait for perfection. We launched so we can learn fast. Phase one is already out there, our B2C direct-to-consumer. We launched via the app stores. We're about to, part of this raise is to kick off our influencer campaign, the rest of our go-to-market. We're going to bring in early revenue and real-world movement data to power the AI and the personalization. This is where we get to crowdsource data. Phase two is when we go into the B2B side. We're going to bring this into the fitness and health industry, trainers, gyms, athletic institutions, wellness clinics, and we're looking to launch that in Q3 of 2025. And then phase three, which is the digital health and clinical integration. This is where we really make the big difference. The first two phases are about really crowdsourcing data and learning and teaching our algorithms and our machine learning and our AI how to create this level of digital healthcare of movement intelligence, and how we can then in that go to market musculoskeletal rehab, chronic protocols. Got about a 24-month runway. Each phase powers the next. More data, smarter AI, and deeper outcomes. And how we're path to profitability, simply recurring revenue model. So on the B2C side, we're going to turn this on in about 60 days, $14.95 per month for the consumer version of the fitness side. On the B2B, it'll be $9.95 per month per client. When we get into recurring revenue tomorrow, that's the digital health. We're looking at about $39.25 again per patient when we work with doctors, physical therapists, and I can give you a whole laundry list of things. This is data-fueled subscription model with exponential upside. Every user trains the AI, making this a flywheel, not a flat line. So, our strategic journey, I think it's important to share that. We saw beyond workouts. We saw the future of care. And like we say, every stage fuels the next. More data, smarter AI, stronger results, deeper impact. When you look at phase one is the data foundation, and that launch, we went live with our MVP on Thanksgiving of November 2024 here in the US. Crowdsourcing movement intelligence. It's launched, it's out there, it's free right now. Like I said, we will turn the revenue on the next 60 days. Phase two is the market expansion. That's going into the B2B side and scaling the reach, the revenue, and the precision. Again, grabbing data, feeding our models with all that data while making a difference in people's daily lives. In phase three, as we said, we're about 24 months from that. That's the digital health intelligence platform. And this is what, again, with this raise, gets us into bringing in the research, learning from the data that's coming in. Every phase stacks more users, better insights, smarter AI, deeper outcomes. This is how we're building our journey. When you look at the competitive landscape, it's interesting. See, in the fitness space, in the health space, every offering out there tries to fit you into a box. And we're made as humanity. We're unique in our body. We're unique in who we are and what our goals are and what we need in life. But fitness apps, they're static. Health trackers, they're fragmented. Nobody connects movement and medicine until now. Personalized like your fingerprint, AI that adapts to every rep, root cause focus, not symptom tracking. Let's get to the problem and solve it and fix it. And with movement intelligence and getting people to move, we're going to not only prevent disease, but help people live longer, better lives. See, we don't fit into a category when we look at all this. So we created our own predictive movement health. The opportunity, we're asking to raise $1 million in our seed round here. As you can see, almost 70% of it goes to our go-to-market to get those users in there, gather that intelligence and revenue. 15% to operations and 16% to development over the next 12 months. You can see where we're going to spend that from a go-to-market standpoint. Assurance factors. I think it's important here. We've hit over 390% of our influencer goal to get our B2C out there. We've already have trade associations, other partners on the B2B side who are ready to go with us. We've launched the MVP. It'll be a full version 1 in April this month. So by the end of this month, we're actually just getting that into the app stores right now. What I'm really proud of is our team. We're not just stars, we're rock stars who've scaled, exited, and rebuilt the future. We've got 8 decades of go-to-market leadership already, 3 decades in the fitness world, 6 decades in health and wellness, 4 successful exits. I've talked about myself, Jason, my co-founder. He's the brain behind the fitness piece of this. Trevor, a great CTO. Hilda, self-made, created her own nutraceutical company from 0, scaled it to 9 figures, sold for 9 figures. So she's an amazing advisor to me, as well as John, who's a retired executive for Northern Trust in the banking industry, corporate governance, and all that. So we're excited for the future. So I'll close with the same question. Did you move today? Your movement, your medicine. Together, let's redefine health. Thank you.
Great. Thank you so much, Scott. That was a wonderful pitch. I'm going to open the floor for questions now. Anyone? Rick, did you have a question?
No, not particularly. I
Sorry.
wondered, can you hear me?
Yes, we can hear you fine.
I was wondered if there was anybody that was remotely trying to create this situation, this model, other than you.
So from our research, the answer is no. Again, every piece is fragmented. There's a lot of stuff going on in the fitness space. There's stuff going on in health and wellness. But what we're doing is bringing that all together. So think about it. You can get a fitness app. You can get a personal trainer. You can use ChatGPT to tell you what to work out with today, how to eat better, and all that.
Mm-hmm.
But imagine a world where you tie into, I've got my Oura ring on, your glucose monitor, you name it. You've got diabetes. One of my best friends, she's type 1 diabetic. She's a nutritionist by trait as well. We know fitness can affect glucose levels. So imagine you have a platform on your phone that can show you how to move today, whether it's an intense workout or just walking down the road, in monitoring your glucose levels and understanding what you need to do. Go to something even deeper than that. Um, Parkinson's, horrible disease, and we're not going to solve that with what we're building, but imagine we can give someone an extra year, an extra 18 months that they can get out of a chair by themselves, that they can walk, play with their grandkids, their kids, and have more quality of life longer. And when you can bring those two worlds together, that's what we can accomplish. And that's what we're trying to do. That's what we believe we can do.
Do you have any idea about what capital may be needed in the future?
Yeah, that's a great question. This is our first round, so $1 million to get the B2C and B2B going. So we start crowdsourcing all that data and revenue. We have some great revenue numbers as well there that gets us into 8 figures. I believe to really build out a few things. One, we want to build out a form of capture technology. Think about your smartphone. It's just 2D, but we can, with AI in training it, get it to see in a 3D model and understand that when you're moving, your elbow should be here, not here, just in a push-up, a sit-up, things like that, how you're walking, how you're running. And our estimate right now, and we're still researching it to be honest, is probably somewhere between 5 to 10 million when we get into really building that type of technology out and seeing how the AI models learn.
There's one more question, unless somebody else has a question.
Please. Thank you. Appreciate it.
What age group do you particularly think this would be the most beneficial for? And you sell the most business?
So, 4 decades and go to market to say everyone is never the answer you're allowed to give. Right. And I get that. But, it's interesting. When we first start out, we were just a fitness app. And then, my specialty, I believe, is seeing the blue ocean, seeing the field. And as we talk to people, we start to see in the B2B side and the medical side, everyone, unfortunately, in humanity is sick. There's, was it 6 in 10 adults in the US are obese, overweight? Are 39%, 6 in 10 have a lifestyle disease. So again, movement intelligence, we can focus on everyone. Now, I also know in a marketing campaign, when we put that, we're going to pick our avatars to go after, figure out where are low-hanging fruits. When you think about 60 and up, which obviously there's a lot of arthritis, diabetes, Parkinson's, diseases like that, we've got to be careful on how do we educate them that this technology is very easy to use. So below those age groups, they're more adapt to using their phones for things. And then you get into the 20 and 30 somethings, they embrace AI. So we understand our messaging is going to be very different for those avatars, but those are all avatars that will use what we have. It's just how and when we go after them.
Thank you.
No, thank you. I appreciate the questions.
Anyone else have any questions for Scott? If not, I had a quick one for you, Scott. Do you expect all your users to kind of have some sort of wearable and or CGM or any other kind of devices for this to work well?
Great question. We would like it. And by the way, we're not looking to build hardware. I know that industry. I don't want to go there. Partner, yes. No, they won't, though. So obviously, the more they wear an Oura Ring, a Apple Watch, and glucose monitor, et cetera, the more data we get, so the more precise the AI can be to help them. But it's not necessary. We can do a lot of great things without that extra data.
And do you have any beta users already?
Yeah, we have about over 60 people using the B2C, which is a fitness app right now. And obviously, we did some testing before that. So we've learned, we already have some UI things we want to change. Like every founder, my baby's ugly, right? So there's things I want to change to make it, from the feedback we're getting, to make it more user-friendly. But again, put it out there and learn. And that's part of this raise, is we can turn on some of the go-to-market spigots, as I like to call them, to start getting, especially with the influencers. We've contracted with 12 influencers, over 4 million followers, to say, hey, go use this. And we've done it from a revenue share standpoint, so it's not costing us money up front. That's the genius from my standpoint, so we can go out there. We've got a couple influencers who have between 3 and 8 million followers who individually who want to partner with us. I want to clean some things up in the UI before I go that next level of growth. because you only get one shot at it. And we know right now we've got to clean up a few things in the UI, just like with every new technology, so that we can minimize churn as we go. Once in the next 4 months we do that, then we'll bring on some of those big influencers.
Great, thank you.
Thank you.
Gabriel, I see you've got your hand up.
Yes, absolutely. Thanks for your presentation, Scott. I have this question of a training set. So I understand that AI is going to be trained to give you personalized recommendations when it comes to your fitness and your physical activity. But how are you going to evaluate the results, and how are you going to train that AI model to give you the best recommendations?
Yeah, that's a great question, an important one. So from the beginning, it's the fitness side. So bringing in personal trainers are going to see. So right now, Jason, my co-founder, he's the fitness brain, as I like to say. He starts you off. One thing that sets us apart from a fitness standpoint is the movement assessment. If you've ever downloaded a fitness app, what does it do? What's your age, height, weight? What are your goals? Three other questions that mean nothing. And here's your workout. As Jason would say, I got to know if you got one arm or two. Can you bend down and touch your toes? So we're training. He's created the movement assessment itself is not proprietary. The way we score it is, and that feeds into the hit. What's proprietary in our IP is how we create movement fitness in your programming. And then as the AI learns himself, and we're building out the team in this part of the raise, to have trainers like Jason seeing how the AI is adapting. Did it go left when they thought it should have went right? So you got to put some humans behind it to watch and monitor it and train it and tell it, no, you shouldn't have done this. You should have given them an extra 5 pounds, not reduced 5 pounds, and here's why. So there's going to be a lot of that has to happen in the beginning. And then obviously, as we get into the medical piece over the next 24 months, it's bringing in kinesiologists, physical therapists, doctors, and again, monitoring all the data we get from glucose levels, how are those functioning. We will build out an advisory team that's going to watch that data and make sure it's adjusting the way we want it to.
Thank you, Scott.
Thank you.
Amazing. I think we have time for one more question. If not, then thank you so
I
much.
still have.
Yes,
I
go.
still have. Yeah. Do you have in your team? I maybe didn't notice or wasn't focused enough. Do you have in your team a data scientist or somebody who is from this background to be able to create this model? As I understand, your app is really focusing on this kind of components, so it has to be good. And I really wonder if one of the founders has that kind of competence.
So we don't have that on our team. That's part of, we get into a deeper conversation of showing you where we're going to build out the team. So we need to bring someone like that in. And then also in the next 12 months, we need to bring someone, lack of a better title, chief medical officer, who can really start building out the digital health and taking the data we have. So those are additions to the team that we do need. And we're starting to talk to people, tap into our network to find those people.
Thank you, Scott. No more
Awesome. Thank you.
questions on my side.
Amazing. Anyone who wants to hear more or talk to Scott further, please let us know so we can
Oh yes,
facilitate.
maybe one very practical question. What is the minimum ticket size for your investment?
So we're
Per
trying to raise
investor?
1 million. Minimum ticket size is 50,000.
60,000.
50,000. And if anyone wants to reach me, obviously you can go to Myworkout.ai, which is .ai. You can also find me at Scott at Myworkout.ai, and our website's up there and everything as well.
Okay, perfect.
Lovely.
Thank you,
Thank you so
Scott.
much.
Thank you, everyone. Really appreciate the questions, the opportunity. Have a great rest of your day.
And you.
Thanks.
All right. Next up, we have Matthew. If you can hear us, are you
Yes, I can hear you fine.
amazing? I'm doing my PhD in cardiac aging, so I'm really excited about this pitch. If you can share your screen, Matthew, and then the floor will be yours. Feel free to give a little introduction, and then we're good to go.
Fantastic. You can see my slide. Okay,
Yeah.
I'm Matthew O'Connor. My friends call me Oki, and I have a platform and a an asset as Cassie alluded to in the cardiovascular space. My background is really in hardcore aging research. I've been working. I went into science to study aging to try to intervene in the aging process, and I've really only ever worked on aging. I've worked on telomeres, muscle stem cell aging, mitochondrial aging, lysosomal aging. I was vice president of research at the SENS Research Foundation. I was there for 9 years, and I discovered this technology during that time and spun it out in 2019 into what is now Cyclarity Therapeutics. I'll skip some slides in the interest of time, and I can come back to things if there are questions. I want to tell an old and new story about oxidized cholesterol. The most common non-enzymatically formed oxidized form of cholesterol is called 7-ketocholesterol or 7-KC. So I'll use those three terms interchangeably. And I started thinking about oxidized cholesterol about 20 years ago when I saw Aubrey de Grey give a lecture about it at my university when I was in graduate school. And I never really stopped thinking about it. And then I got the opportunity to oversee research on the topic when I was at the foundation, and eventually got frustrated at the slow pace of it and decided to do something about it myself. And then it suddenly took over my life. So here I am. So in the 1990s, there was already this mechanistic idea that oxidized cholesterol might be causing foam cells. Foam cells are macrophages that get recruited to the site of plaque in your blood vessels to try to clean it up. And macrophages are just supposed to eat up junk. And when they come to these sites, they do exactly that. But when they eat the oxidized cholesterol, they can't digest it and they can't export it and they can't use it, and it's very toxic. And it ends up shutting down their ability to metabolize. And they just keep eating as they turn into these big blob cells called aptly and descriptively named foam cells, which never go away, kind of like senescent cells. Uh, and they become a part of the problem and a big part of the mass of the plaque. And kind of like the fireman turning into the arsonist. What we and others in the sort of an emerging field of cardiovascular are trying to do is seeing if we can get macrophages back to work to clean up the plaque. And so what we set out to do was, well, to ask the question, if we get rid of the oxidized cholesterol, will foam cells go back to being macrophages, and can they get back to doing their job? And I'll just come back to this slide for 1 second and point out that while it was, the in vitro evidence in vitro and mouse evidence for oxidized cholesterol causing cardiovascular disease started to emerge in the 1990s, it was in the last decade that we've started to see convincing clinical data that oxidized cholesterol in your blood is associated with incidence and severity of cardiovascular disease, suggesting that it's causative and not just correlative, as well as I think rising it to the level of being a good drug target. I should say that we came at this from the perspective of this is something toxic that accumulates with age in various cells and tissues, and that we should get rid of it as an aging target, and eventually settled on atherosclerosis, the plaque in the arteries, as an initial indication, but that we think that there's other good indications, chronic disease indications as well, and I can touch on that later if we have time. So I went back to the literature to see if there was a potential hack of something that I could use to get rid of the oxidized cholesterol. And I discovered these little molecules, not that little, these cyclic carbohydrates, these macrocycles called cyclodextrins, and they're microbially produced structures, and they've been used for about 80 years in science and industry, for industrial solvents. Now for in medicine, they're used mostly to deliver drugs as excipients. There's a couple approved, and those started coming out 25 years ago. And they're some of the most common excipients used in medicine now. They can be made to be extremely safe, and the regulators are, because of this, are quite familiar and comfortable with them. There's only one drug based on cyclodextrins yet on the market. It's called Bridion. It's owned by Merck. I believe it sold 1.7 or 1.8 billion last year. And what it is it's an antidote for an anesthesia agent, and it works just like our drug is built to work. It grabs onto a paralytic called rocuronium that's very commonly used in surgery, and it immediately competes it off of the receptor that it's bound to, and floats away with it, and then you pee the whole complex out. You wake up right away from your surgery with fewer side effects from the anesthesia. It's kind of a miracle drug. It works extremely well. It's done extremely well commercially, and that's because the patients are just better off for having had it. There's no real side effects at all from taking Bridion, just from the paralytics that you get less from. So for the drug that we've invented, what we did is we did computational chemistry to model the oxidized cholesterol, which you can see here in yellow in the cavity here. And we did computational chemistry to model which size and shape and which engineering of the cyclodextrins, the side chains and whatnot, would be ideal for binding with high specificity and affinity for our target, the oxidized cholesterol. And a lot of the core of our intellectual property is around this dimerization of cyclodextrins in the right configuration and structure to bind the target, and we own a lot of that the space around cyclodextrins, around engineering them in with this kind of strategy, not just for this one drug. So I'll touch on just a little bit of biology, and I can answer more questions later. So we set out to try to take foam cells and turn them back into healthy macrophages, which, as far as I know, has never been demonstrated before. And now we are getting ready to publish this in a prestigious cardiovascular disease journal. So here's the macrophages. We give them just a little bit of the oxidized cholesterol, and they balloon up into nasty foam cells with this red dye is what demonstrates it, those lipid droplets all over the cells. And then we give them our drug, and they go back to being healthy macrophages. Not only that, like I said, there's a whole manuscript behind this. They go back to doing phagocytosis, which is their main job. Their gene expression profile goes back to normal, and they're back to being healthy macrophages. In vivo, we recently accidentally discovered something pretty cool that's going to help us in our clinical trials that are running right now. So what we found is that even in young healthy rats, that this urine test that we're going to be using in our patients is actually very effective in any animal, and it works extremely quickly and extremely sensitively. And so this means that in the clinical trial that we're running right now, we're going to get lots of data from the phase 1 healthy volunteers. This is being run in Australia under Dr. Steve Nichols, the foremost expert on plaque in patients, and he's a world-renowned cardiologist who's run many of the biggest clinical trials on some of the biggest cardiovascular disease drugs that are out there. I'll just say briefly about the trial. We have started phase 1. It launched in January. We're finishing the second cohort of patients now. There's quite a few cohorts, so this is going to run for most of the year. We have permission to go all the way through patients. We have a small cohort of 12 patients that will be starting late this year in Q3 or Q4. That will be getting the dose that we discovered, the highest dose we discovered to be safe. And then next year, we plan to move into phase 2 in 150 coronary artery disease patients, which is the same type of patients that we're using in the later part of our phase 1. Our world famous clinical advisory board, all famous cardiologists, our board of directors, are the money that we are raising right now. So we closed on $6.4 million in December, and we have a term sheet for to close on at least 2.6 million and up to 5.6 million more. We have a little over 2 million of that committed currently, so we need to raise at least another 600,000. We have as much as 5.6 available total, so 3.6 more. Timeline of events, launching phase 2 middle late of next year. Finishing all the dosing of phase 1 before the end of this year. We'll have all the safety pharmacology work done by the end of this year, and there's some follow-up stuff in the patients that will go into early mid next year. But everything that we need to launch phase 2 will be done before the end of this year. We have this amazing computational chemistry platform that that we now have layered on machine learning and AI, so that we can take the human out of the equation of designing the cyclodextrin. And there's many other potential applications of our technology, including anesthesia reversal, like Bridion, bisretinoids for macular degeneration, nanoplastics, all things that we're looking at. And like I said earlier, in chronic disease, oxidized cholesterol is also implicated in Alzheimer's disease, stroke, macular degeneration, and liver failure, many of those of which we're looking at now. So in the interest of time, I'll stop there and take questions.
Thank
Where do you
you
get your
very much for
patients
that
from?
lead.
Where do your patients come from?
They're from a cardiologist in Adelaide, Australia, from their practice. The patients, the healthy volunteers, are recruited by the CRO that's running the trial, again in Adelaide, Australia.
Why would a patient decide to use your process?
Why would they plan to do it?
Yeah, you're doing kind of an experimental thing now, and are these patients have a significant cardiology situation?
Yeah. Yeah, the basic reason, and the basic reason for our approach and for our technology is to reverse plaque. Currently, there's effectively no way to do it short of surgery, right? So you can have plaque cut out surgically, you can have bypass surgery, you can have stents put in, but as far as non-surgical interventions, there's all the lipid-lowering medications that are currently on the market just slow down the accumulation of plaque. They don't reverse it. We are trying to reverse it.
Thank you.
Cool. Anyone else? Yes, actually, Alex.
Thank you, Matthew, for the presentation. I had a couple questions, actually. First of all, how does the cyclodextrin actually enter and exit the macrophages?
We don't exactly understand the mechanism. In that case, we haven't drilled down into that. It's not something that the regulators particularly care about. I suspect there's a couple of different mechanisms that cyclodextrins have been shown to enter various cells, phagocytosis and receptor-mediated endocytosis. I suspect in this case that it's non-specific phagocytosis, but we haven't chased that down yet. And it's not a top priority. I'd really like to know as a scientist, in terms of practicalities of getting the drug through trials and approved. It's as far as we can tell, not very important to know.
But you are sure that it is removing 7-ketoglycerol from within the macrophages rather than from the environment around them?
Yes, actually, that's a good question. We have measured that many times over very sensitively. We've published that already, and we're publishing more about that now. So yes, we have definitely shown that it is removing the oxidized cholesterol, 7-KC in particular, from macrophages. We've also shown it in ex vivo in plaque tissue from patients that we've soaked in our drug.
Right. Okay. And you mentioned that you've seen the morphology of the macrophages go back to a more healthy state. Have you done any functional assays to assay whether they're behaving as normally functioning macrophages should in that context?
Yes, the main thing that macrophages are supposed to do is eat. That's phagocytosis. So we can run very standardized phagocytosis assays. When they turn into foam cells, that eating goes down. And after we treat with our drug, we see them, and we see them clean up. They go back to eating. So that the main job activity goes back up the way it should.
Okay. And
And
sorry, last
that's
question.
what we're publishing now. Yeah.
Yeah, last question. Did I get it correctly that you mentioned that the 7-ketoglycerol that you see increasing in the urine excreted was in young rats?
Yeah, that was in young rats. I didn't expect to see that. We were just doing that to test our urine test that we're going to be using on the patients, and we accidentally discovered that the test is so sensitive that we can even detect a little bit from young rats, which makes me realize I thought we weren't going to get any target engagement data until we got to patients at the end of this year, and that we would only get a tiny bit of data from the 12 patients. But now I realize that it's going to work on the 72 healthy volunteers that are going through the dose escalation right now.
Yeah, that actually surprised me in two respects. First of all, because you saw it in young rats, which I assume don't have that much plaque. How young were these rats? Were
None.
they?
They were a couple of months old. I don't remember
Yeah,
exactly,
okay.
2 or 3
So
months
really young.
old. Yeah, super, really young and healthy. So it's not going to be as much as you would see in. So these animals don't have any plaque. They weren't even on a high-fat diet. Uh, it wasn't it wasn't
Yeah.
any kind of intervention trial. It was just testing the urine test, and we accidentally found that in a control, which turned out to be
Yeah.
something of a mini breakthrough for us.
And actually, it didn't even seem like that little of an amount because, as far as I could see on the y-axis, maybe I got this wrong, but it seemed like nanograms per microliter. When you scale that,
Mhm.
that's on the order of milligrams per liter of urine, which kind of seems a lot for a oxidized cholesterol product.
Yeah, we haven't fully chased down like how it would compare to old animals. What we haven't chased this down at all. This is very new data, so we don't know how that will compare to old animals, to animals with heart disease. We're working on that now. But I think part of the reason that you're seeing it like that is that you're seeing it getting very concentrated very fast because our drug acts really quickly. Our drug
Mhm.
isn't Drano, right? So it's not going through and like blasting through and cleaning everything out in real time. It's just targeting the oxidized cholesterol and then allowing the plaque to heal slowly. But it does its action of grabbing the oxidized cholesterol within 15 minutes. So it comes to equilibrium with whatever environment that it's in within 15 minutes, and then it's excreted with most of it is excreted within an hour. And so the PK and the PD, which I didn't show, actually overlap each other basically perfectly. And so they're coming out together all within an hour in the rats. And so I think it's getting concentrated very quickly. And that's why you can see a significant amount of it there.
Okay, okay. Oh, thank you very much.
Go
That's a
ahead,
good
Gabrielle.
question. Nobody's ever asked me exactly that before, but that's a cool question.
Yeah, I'm trying to understand the mechanistic steps in the process. So what I understand is the macrophages are supposed to get into the arteries to collect the oxidized cholesterol, and then they're supposed to get out, but they can't because they get stuck inside, right? If you
Yes.
with your molecule, you would get the macrophages healthy again because actually the oxidized cholesterol is excreted out of the macrophages through this cyclodextrin. But don't they remain stuck in the arteries, which means that the reason why the macrophages in first place entered and tried to collect them is compromised?
Um, So I think the answer, I'm not sure I totally understand your question, but I think the
Yeah.
answer is that since we reverse their phenotype and turn them back into healthy macrophages, they should be able to go back to work and clean up the plaque. Now, what macrophages are supposed to do once they're done doing their job is kill themselves and float away. So that's what we would hope would happen on a mechanistic level. We haven't, proven all of those steps yet, but that's what we expect to happen.
Okay.
Another
And
question.
another.
Well, excuse me.
No,
Wait.
please go ahead. Go ahead, Rick.
Well, I was going to ask, are there any regulatory hurdles that you haven't cleared yet or you're on the way to clearing?
Yeah, good question. So we've gotten the equivalent of IND in Australia, of course, because we've launched there. We've had a pre-IND meeting with FDA and equivalent meetings, scientific advice meetings in the UK, and we'll be filing for IND with FDA later this year. We are still working on exactly which other environments to go into, considering UK, Brazil, and Europe with the EMA. But probably not all of those because it would be too many, but probably one of those three in addition to Australia and the US for phase 2.
Thank you.
Anyone else have any questions? If not, I've got a few I'd like to ask.
Yes, one other question.
Go ahead,
What
Rick.
do you anticipate the situation would be with an insurance company that's covering this as a benefit?
Yeah, I mean that's the path, whether it's a private insurance company, as we have mostly in the U.S. or with U.S. Say like Medicare, or in countries with nationalized healthcare. We expect reimbursement to work similarly. That that we'll have an indication, and that there'll be a value proposition, and that there'll be a diagnosis for, say, coronary artery disease. There's a number of atherosclerotic sub-indications that we think we could be looking at. We could be looking at angina, the chest pain caused by atherosclerosis. So atherosclerosis itself is generally not a diagnosable. Well, it's a diagnosable condition, but it's generally not a treatable condition. So a specific indication like coronary artery disease, people, post event, like post a heart attack, post stroke, maybe people with carotid plaque, although surgery is quite straightforward in those cases. Those are some of the sort of lower hanging fruit indications. Peripheral artery disease is a tricky one, but also something that we think could be quite effective with. It's tricky to get approved, not necessarily to treat. We think it would be great if we could get it approved for peripheral artery disease as well.
Okay.
Would you have to target your drug specific to like a cell type, or do they have some sort of mechanism to know exactly where to go?
Good question. We have not engineered our drug to have a preference for macrophages or for any other cell type. We're delivering it non-specifically in any place that it can penetrate through the bloodstream. It will go. We haven't yet chased all that down yet. We know that our drug doesn't bioaccumulate in any organs, but that doesn't mean it doesn't get into the liver, the kidney, it probably doesn't get into the brain. But other than that, it should be able to distribute widely. We're interested in targeting technologies and targeted delivery. We're interested in collaborating with companies that have targeting technologies, but we're not currently working on any.
Oh, and then I just had a quick question about the kind of algorithm that you guys are using. Is that mainly to find derivatives of your drug, or is it to like kind of adapt it to specific various other targets as well?
To create new, fundamentally new drugs of the same class against different targets. So A2E, the primary bisretinoid that accumulates in the macula during the dry form of macular degeneration is another target, for example, that we're looking at, and it would just be a different cyclodextrin engineered very specifically for that target.
Amazing. I think that's it from my end. Does anyone else have any final questions before we wrap up the session?
I can go with one final one. Matthew, do you know whether it also removes 7-keto cholesterol from cellular membranes? Because I imagine it can accumulate in those as well.
We believe that it does. So we don't know exactly how much of its activity is happening at the plasma membrane. So it should accumulate in the plasma membrane. It's known that 7-KC oxidized cholesterol is known to damage plasma membranes, and that's part of its toxicity. So we expect it to be pulling out of the surface of the plasma membrane. We don't know how much of its activity is there at the surface versus in the cytosol at the lipid droplets versus, say, getting into the lysosome, which in the case of macrophages, they have these enormous lysosomes where you would expect the 7-KC to be ending up. Those are all really interesting questions that we haven't tracked down yet.
Well, sorry, just follow up. Could you potentially tag the 7-KC and see where it's localized within the cell when it's taken up?
We could. It would be a little bit tricky, if it's a bulky tag, it's going to impact its, if it's a fluorescent tag, it's going to be really bulky, and then, that's going to influence how it behaves. Subcellular uh, radiography, I think, is a little tricky, but that would be the best way to do it. That's what I would ideally like to see. The other thing we could do would be to like tag it or just we have pretty good technology for measuring it. And so even without tagging it, we can feed a bunch of it to cells and then do say a subcellular fractionation and say how much it was in the plasma membrane, how much was in the lysosome fraction, stuff like that. That might be the easiest way to answer that question. Not super easy, not super cheap, but something I'd like to do.
Yeah, not that it's critical for whether it works or not. It would be interesting to know what the mechanism is, where it's actually
Yeah,
removing most of the 7-KC from. Yeah,
yeah,
thank
I
you.
agree. Yeah, always so much more to do.
Did you see any improvement in cardiac function in any of your animals?
Our My position is that the short answer is yes, but my position is that these mouse models of atherosclerosis are actually holding up the field. They're really artificial. These complete knockouts. The two common ones are ApoE knockout and the LDL receptor knockout mice, and they're both really blunt instruments. They completely knock out the ability to either internalize cholesterol or export cholesterol from cells. It's very unrealistic. It doesn't happen in humans. And we do know that our mechanism depends on on reverse cholesterol transport to happen. And so if you completely knock out the ability of RCT, and we know that our drug treatment, I didn't mention this, increases reverse cholesterol transport in the macrophages, when we're delivering our drug. We're publishing that now as well. So, the most popular one being the ApoE knockout mouse, we showed some effectiveness in that, but I don't even know if I believe our own data on that. Others have published kind of the same thing, and others have had a hard time repeating it. So the best models for atherosclerosis are pigs and primates, and it's honestly nobody keeps colonies of like old, chubby pigs and monkeys around waiting for people like me to come along and want to pay to use them. But we do keep colonies of humans around doing that. So it's actually cheaper to be testing this in humans than in animal models, I believe.
Amazing. Thank you so much, Matthew, for the amazing pitch and interesting questions as well.
Sorry,
Um,
one final question came up from what Matthew said. You mentioned that it's dependent on reverse cholesterol transport. Why would that be if you're actually removing the oxidized cholesterol directly?
Uh, okay, yeah. So there's a subtlety there. It's not dependent. Removing the oxidized cholesterol is not dependent on RCT, but resolving the foam cell phenotype, then what we do is we take away the oxidized cholesterol. For some reason, that's inhibiting a number of lipid processing processes
Right.
in the macrophage, and all those get turned back on as soon as we remove the oxidized cholesterol. So the overall mechanism is dependent on RCT. But removing oxidized cholesterol, you're right, is not
Okay,
dependent on that.
got it. Thank you.
Thanks,
I do.
Alex. And yeah.
I do have a last question. We still have 2 minutes. You talked about the molecule you're working on, and you've also talked about this AI algorithm that you have that allows you to play with how to fold the molecule to make it work. Is this is some sort of a side project, or do you have an asset model for this, a business model, or have you just built this for the sole purpose of creating this molecule? Well, how do you go about those two different assets that your company has?
Well, yeah, I'd say that to be fair, most of the current, the present value of our company has to be considered wrapped up in our primary asset, this clinical stage asset. But we invented our drug with computational chemistry, and then we continued to build and improve on that platform. We've published several papers on it. It's a very niche area, but we're the best in the world at modeling cyclodextrins and their activity. And so we're continuing to develop that technology, both the base technology of the modeling and trying to develop and test prototypes for other targets, some of which I mentioned, like anesthesia reversal, which, so to give an example of a value from that, I think that we could be selling an asset to a pharma company that could be developing it because there's a very clear path for developing an antidote to anesthesia reversal. If we had proof of concept in a mouse, say, you wake up a mouse faster, I think we can do that in a year and a half and sell it to a pharma company for $50 million. That's some of the value of the platform.
Interesting. Thank you.
Mm-hmm.
Great. Let's end it on that note. If anyone is further interested in speaking to Matthew, please let us know. Yeah, thank you so much for attending. And Matthew, thanks for the lovely pitch. We'll see you guys in a couple weeks and have a great rest of your weeks.
Thanks so much, everybody. It's been a pleasure. Great
Thank you.
questions.
Bye bye.
Bye-bye.